Hi class, Dr. Jim here. In this lecture, what we're going to be looking at is a process called transcription. And this is an important step when we want to make proteins in our body. As I talked about in the last lecture, DNA is the instructions, the recipe, the blueprint to make everything in your cells. Okay? In order for those things to happen, you need this process, what is called transcription and translation. We're going to talk more about that today. So hopefully you watched the previous lecture on the DNA structure and replication, another important process. But in today's video, we're going to look at the transcription process and then focus in on translation. So what are the objectives here? Well, the objectives are, first we're going to figure out what is the central dogma of biology. This is an important process in which we go from DNA to RNA to proteins. And this is an important step because we need this in order to make proteins. We have these things called genotypes and phenotypes, and we're going to look at that more when we get into our genetics and talking about blue eyes and brown hair and things like this. Now you can see I have blue eyes. Maybe you can't see it, I don't know. But blue eyes is a physical appearance. That's my phenotype. And in order to see those blue eyes, I have DNA coding, which is called my genotype. My DNA codes for my blue eyes, and then the proteins that get made off that DNA are expressing or showing the blue color. If you have brown eyes, you have the DNA that says make brown eyes, and then eventually your proteins make the brown color that you have in your eyes. Okay? And that's kind of how this works. And so what we're going to be looking at is the process, the step-by-step process that takes place. First step is going from DNA to RNA. The second step is RNA to protein. Okay? The second thing we're going to be looking at is the process of transcription. Now, I wouldn't say this is a hard concept, okay? What I would say is that it's easily confused with the second step. That's why I broke this down into two videos. Not only for the length of time and give you more time to kind of, swallow this all in and kind of process it. It's just so that you can kind of keep these two things straight. Transcription is the process or the first step going from DNA to RNA. So we have our DNA, our transcript, and then we go into our RNA, which is the copy of the DNA. And we're going to talk more about that. In the next video that I have that accompanies this, we're going to talk about how the mRNA then gets converted into protein. And that's the important step, and that's called translation. And again, because I think they both start with T's and they seem very similar in how they sound and pronounce, I think people get really confused. And hopefully after watching this video today, along with the second video that I have, the translation, you won't be so confused in how to keep these things straight. So let's move on and we'll figure out if this will work out for you. Okay. So in order to get that genetic code of life, we have the mechanism known as gene expression. I kind of briefly talked about this. Me having blue eyes, what does that mean? Well, since I have blue eyes, I have DNA inside my cells that say make the protein for the color blue so that my eyes are blue. Okay? Has that instructions. In order to get those instructions into the final product, which is protein, it needs to go through this thing called gene expression. So what that means is my gene has to be turned on, and basically make a transcript of it, make the copy so that it then goes to the worker. The worker then takes that message to the ribosome and makes the product. Okay, and that's kind of what you do when you're building a toy or making a recipe. You are that worker. You are the mRNA, taking the instructions, following the instructions, and then making that final product, which is the protein. And that's basically what we're talking about here, is that DNA has the information, but DNA doesn't get expressed in structure and function. It just expresses the instructions. Okay. Then the expression into structure and function requires the RNA, the worker, you making the cake. Okay. And then finally, once you make the cake, you have that final product, which is called protein. And we're going to talk all about that in this lecture. Okay. Or in the next lecture, I should say. So what is the central dogma? The central dogma is probably the number one thing that you should get out of biology. Anything else that you remember about biology, you should know the central dogma. And the central dogma means from DNA to RNA to protein. Every cell follows this mechanism from bacteria up to humans. So this is what is required in order to make anything inside the cells. And so that's why it's important to know. DNA is the instructions. The RNA is the worker that takes the instructions, makes the product, and finally the final product is the protein. And you can kind of see it here. Again, we have our DNA, the double-stranded DNA, okay? We have these two different strands. And again, we're going to talk a little bit more about what non-template versus template is in just a minute when we talk about transcription. And I'll come back to those terms in a little bit. We then have the mRNA, which is basically a copy of the DNA. You can see up here, they look very similar, okay? Or actually, in this case, they are the same. The only difference is that U's get replaced, or the U's replace the T's. And this is a bad example because there's no T's in here, so you don't see that example. But we'll get into that in my example. I have lots of T's and U's and that stuff in just a minute. The mRNA then takes that instructions and goes out to the ribosome and does the process called translation. And what happens is you get this protein that gets made. And we'll get into that all in the next lecture. So make sure you watch that next lecture so that you know the difference between transcription and translation. We're going to look at translation next time, and that's converting the RNA or nucleic acid code into protein code. So very good video. I highly recommend. Make sure you watch it. But for today and for now, we're going to just focus in on the process of going from the DNA to the RNA, and that's called transcription. Okay? All right. So to understand the genetic code of life, we need to know what RNA is. And I know in Chapter 3 we briefly discussed a little bit about the difference between DNA and RNA. But now it's really important that you understand the differences because there are some differences between DNA and RNA and what they look like and what they do. RNA is a polymerase of RNA nucleotides. And if you remember what a nucleotide is, it's basically a phosphate, a sugar, and the base. So you have a phosphate, the ribose sugar, which is an RNA, so I can't really do, that's not bad, ribose sugar, and then the base, which is either an A, U, G, or C. Now again, that's one of the differences. So here are the two differences that you need to know. First, RNA uses ribose, okay, and the difference between ribose and deoxyribose is one oxygen. Okay, in ribose you have the oxygen, in deoxyribose you don't. Okay, and that's what deoxyribose means, is that you're missing the oxygen. The second difference is that U replaces T, and that thymine and uracil are very close cousins to one another. The only difference is thymine has this methyl group where uracil does not. And the only reason for this is that uracil is less stable, it can fall apart easier, and there's a lot of reasons why you want RNA to be less stable, and we'll get into that later on. If you have questions about that or want to know more, just ask me in the lab, and I'll be glad to tell you all about it. Okay? The other thing is we have three types of RNA. Two of them we'll learn next time, but the third one is very important for transcription, and that's the messenger RNA. It basically makes a message of the DNA that you have in your nucleus. It makes and copies it word for word, and then uses that copy to make the protein. Okay? That's the messenger. The other two are called rRNA or ribosomal RNA, and we see that with the different subunits of ribosomes, and we'll talk about that next week, or in the next lecture. And then also the tRNA, which is the transfer RNA, and this is important for bringing amino acids over to the ribosome, and that's really important as well. And again, these two things, the ribosomal and the transfer, we're going to see those next time in the next lecture. So keep them in the back of your head and say we're going to look at those next time. What we're going to focus in on today is the messenger RNA. Messenger RNA is the important step in transcription. Okay? And so if we look at the structure, again, the nucleotide is the phosphate, the sugar, and the base. That's the important thing. Phosphate, sugar, base. Okay? And that is a nucleotide. And you can see that RNA lines up just like DNA does with the phosphate, sugar, and base. However, you only get one strand. RNA only makes one strand. It doesn't make two like the ladder that we see with DNA. And that's another difference that we see between RNA and DNA. RNA is always single-stranded. Okay? So if we look at some of the differences, again, RNA has ribose, DNA has deoxyribose. Bases include all the same bases except in thymine is in DNA, uracil is in RNA. And then finally, the strands, we see single-stranded in RNA, double-stranded in DNA with the base pairing, and then we don't see a helix in RNA, which we do in DNA. So make sure you know the differences, because we like to ask questions about the difference between RNA and DNA, just to make sure you're paying attention and you know what you're talking about a little bit on the test. So make sure, good slide right here for the test to study for. Okay? Now the process of transcription is basically taking your DNA and making RNA. Now, the way I like to explain it to my students and what transcription are, and since you're my students this semester, you're going to hear my story. Think about it, you miss class, and you have to borrow your friend's notes in order to copy them, okay? You take his notes or her notes, and you make a copy, word for word. You're writing them all out, okay? That's called transcribing. You're making an exact copy of your notes from your partner, or your lab partner, or whoever, okay? Make that copy. But let's say you copy it slightly different. Let's say your lab partner or the person you copy from writes in cursive, but you print everything, okay? That's the subtle difference, and that's kind of how we look at RNA and DNA. They're very close, but there's subtle differences between each other. Maybe have different handwritings. You can use any example in this case. So what you're doing in this process is basically making a copy of that DNA. You make one strand of RNA that's a copy of the DNA, and you use that to then make your proteins. It basically is making a copy of the instructions to then go make the protein that you need, and that's called translation. We'll look at that next time. So transcription, making a copy. So think about that, making a copy with some subtle differences. The two big differences is that C and G go together, again, GC and CG, and that in this situation now A's go with U's, and likewise U's go with A's. There's no T's in RNA, and so that's how you always know when you're dealing with an RNA. Look for the U's, okay? Once you're done, you get what is called a pre-mRNA transcript, and we're going to talk more about putting these things together and making a mature mRNA transcript in just a minute, but this is important. So right as it comes off the DNA, you make a pre-mRNA, and then it gets processed to make the mature RNA. And this only happens in eukaryotes, cells that have a nucleus. In prokaryotes, as soon as it comes off the DNA, you're ready to make protein. So subtle difference, and again, we'll talk about that in just a minute. So here's my visual for you. If you look at the top strand, here's the top strand of DNA, and remember 5' 3', and the only reason why I bring it up is because DNA is always written 5' to 3'. The bottom strand is always written in the opposite direction, okay, bottom strand of DNA. During the process of transcription, what happens is that your DNA that you have opens up, and then the RNA polymerase will bind onto the bottom strand, okay, so it actually binds onto this guy right here, this bottom strand, and reads it from 3' to 5'. And the reason for this is then it can build the top strand, which looks like an exact copy of the top strand of DNA here. So if you really want to be cute and just remember how to make mRNA, it's basically this top strand, except the T's are replaced with U's. Okay, and that's the only difference. So if you look, if you compare this, the red line to the top one up here, you say, wow, this is exactly the same thing, except the T's are now U's, and now I have my RNA. As soon as that gets made, the RNA falls off. That goes over out of the nucleus to go to make protein, and then the DNA comes back together to form the double helix. Okay, and that's really the process of transcription. You basically open up the DNA, make the transcription with the bottom strand, make that copy, the RNA comes off, and the DNA goes back together again, and that's the process of transcription. Pretty easy, not too hard. And again, I'm not going to ask you every enzyme involved and everything else. I think you should know RNA polymerase because that's an important enzyme to make RNA. But really, I don't care if you know RNA primers and all this other stuff and how the DNA opens and all that other. That can be saved for a molecular biology class. What I want you to do is just get the concept. Concept is really important. What happens during transcription? If I asked you what happens in transcription, you can tell me, well, that's where the DNA opens up, RNA is made, comes off, the DNA goes back together. Okay? You make that copy, and that's what happens. All right. For further video watching, I have this up on my YouTube channel. Make sure you watch this video. It's a really cool computer graphic. It's about 5 minutes long, maybe not even that, 3 minutes. And it just shows you the process. Again, the DNA opening up, the mRNA coming on, making the transcript, falling off, DNA going back together. And just takes you step by step through the process. And so I think it's a really good video. Definitely watch this video so that you get kind of a visual idea of what's actually happening. All right, here's the picture. Again, DNA opens. Here is my non-template strand, which is on the top. Here's my template strand, which is on the bottom. And you can see here's the RNA polymerase going along, chugging along and building my RNA. And they show it as that basically the RNA is briefly attached and then slowly just falls right off, and that continues to go on until you get to the end of the DNA, the terminator part. And again, there's lots of steps involved. I'm not going to worry about the steps. I just want you to know the process. Okay? You can read all of this, but basically it's the same thing that I said before, in that the DNA opens up, your RNA polymerase binds onto the bottom strand, that template strand, reads it, builds the mRNA on the top. Instead of T's, you get U's. Once that happens, that falls off. The DNA comes back together, and now you have that mirror image of mRNA that looks just like the top strand of the DNA. And once that happens, it gets processed, and then it gets made. So again, you can read all this stuff and go through it all and see. It probably confuse you more. I just put it up there so that you guys have it. You know, read through it, but really it's as simple as I'm talking about. It opens, makes a transcript, falls off, closes. Okay? And here you can see RNA polymerase, and we can have multiple polymerases happening at one time. So it's not just a one and done type of thing. You actually have multiple transcripts, especially in transcripts that make lots and lots of really good proteins. Okay? When your liver is making lots of proteins or your kidneys are making lots of proteins, you need to make lots of transcripts really quick. Okay? And this process goes on and on. And if you watch that video that I have on YouTube, I think it talks about how long this process, and that in your bone marrow cells, you're making millions of transcripts per second. So think about that, how many transcripts you're actually making in an amount of time. Now, in eukaryotes, your RNA gets modified before it leaves the nucleus. In prokaryotes, no nucleus, no modification. It goes and gets made into protein right away. Eukaryotes, since you're in the nucleus, you actually get a couple of things. The first thing that happens is that you add a cap, like a baseball cap. It adds the cap to the top, and that kind of protects it before it leaves the nucleus. The second thing that it does is it adds this long string of A's across. So think of Fonzie and A. Adds a long set of A's, and what that does is basically tells the ribosome, this is where you fall off at. When you see these long stretches of A's, that's where you come off. Okay? You fall off at that point and you stop. And so that's an important addition to the mRNA transcript. The other thing that's important in the mRNA is that you actually have two types of DNA in your cells. You have what is called exons. Exons get expressed. So always remember exon, EX for expressed. That's the one that gets made into protein. The exon is what's made. The intron is what they used to call junk DNA, but we actually have found that it's more important than just junk DNA. There's a lot more to it than that. There's a lot of regulation and other things. But just think it's DNA that's not needed to make the protein. Okay? That gets clipped out before you go to the ribosome. And so this is called RNA splicing. We have little molecular scissors in our nucleus that cut the mRNA to make the full transcript. And once we cut the introns out, we put it all together and make what is called the mature mRNA transcript. Okay? And I know there's videos and other things online to kind of show you, and in this next slide, I kind of show you in the picture. Here's the DNA with the exons and introns. You do transcription, which makes the mRNA transcript. Okay? You have this pre-RNA. You add the cap, which is like the baseball cap to protect the RNA, and this long A tail. Remember Fonzie, and that. You make this long mRNA, and then what happens is any intron gets clipped out. Here come the scissors, cuts out the introns, and what you have left is the exons. The exons are the coding regions, or the ones that express. And that makes the typical mRNA, and that's what goes to get made into protein. That leaves the nucleus and then gets made into proteins. Okay? So to summarize, the central dogma of biology is DNA to RNA. That is transcription. That's what we talked about today. And the next step where we're going to look at is RNA to amino acids and proteins. That's translation, and that will be in the next video. So make sure you watch that next video, and taking that mRNA as it leaves the nucleus and then makes the protein. Okay? Transcription, you know the messenger RNA is made by RNA polymerase, and that U's replace the T's, okay? And that eukaryotes, we process. We have to clip out the introns and we add a couple of things to the ends. Prokaryotes, that doesn't happen. As soon as the mRNAs get made, they get shipped out of the cell, and that's the important thing. Okay? And with that, I've come to the end of this lecture. If you have any questions, please send me an email. Please ask me in class. I'll be happy to help you on any of those things and kind of explain it, draw it out. In lab this week, you get to build these molecules, and I think it will make a little bit more sense once you start building these things. With that, if you have any questions, please ask, and I'll see you next time.